Hi, so my final proposal is the use of degradation tags on ARA-H1 protein from the peanut to inhibit interactions with IgE binding, which is the usual cause of allergic reactions. So why do we have allergic reactions? It's basically an immune response facilitated by the binding of immunoglobulin E, or IgE, antibodies to its specific target. In this case, it's an allergen protein. When these two bind together, they will then bind to the mast cell and release compounds like histamines. This is what causes inflammation and the usual allergic reaction symptoms like rashes, hives, itching. And then these symptoms can vary depending on severity. There has been some past research done on allergic reactions and how to decrease the response. Peter Beetham wanted to generate a non-allergenic peanut. He hypothesized this peanut using gene repair oligonucleotide, or GRON, I think. It would modify the structure of the surface of the peanut versus cutting out certain proteins, which would then result in something that is not considered a peanut anymore. So modifying the surface structure would inhibit binding with IgE. So ultimately, he wanted to eliminate any undesirable function of a protein while preserving its original structure. So the mechanism of GRON, when it detects or recognizes a DNA sequence that would encode for a protein that would cause an allergic response, it would prompt something like a spell check, if you will, and use plants' native repair mechanisms to change that sequence up a little bit, and then therefore binding is not possible anymore. Another study I found used RNA interference to silence ARA-H2 protein. This was done in the Dodo lab, and they initially tried to create fertile transgenic plants that had silenced genes, and like their first attempt, it didn't quite work, or like there was something that was wrong with it. So in the paper that I read, they had fixed and adjusted whatever they did in the last experiment, and they were able to get fertile transgenic peanut plants that had a silenced ARA-H2 protein. The whole study, they tested these plants to ensure that that protein was in fact eliminated, and then at the end they gave these peanuts to allergy patients, and they found that there was reduced binding capacity for IgE. So that meant that this pathway might be a fruitful area of further research. Other possible solutions would be targeting the host instead of the peanut plant. This could be done probably through like DNA or protein vaccines or immunotherapy. So why is this important? There is currently no allergy cure. It's usually just something that's managed by the individual. They just avoid specific foods, and then that can get tricky depending on how bad their reaction is. It's estimated that 200 deaths per year in the US are caused by fatal anaphylactic shock. And so allergic reactions are not only a health issue, but it can affect things like the environment, economy, society. A study showed that peanut allergies caused increased medical costs and decreased quality of life for those who have the allergy and their families or caregivers. For childhood food allergies, it costs the US about $24.8 billion per year. So finding a cure to an allergy would greatly help alleviate these challenges that the individual has with their health, and then also just the country as a whole. This is my general overview of how my experiment would be set up. Hopefully it's somewhat clear, but For the first step, I would take or extract my gene of interest, which would be the ARA-H1 protein, extract it from the peanut plant, and then use fusion PCR to attach a SSRA degradation tag to the DNA sequence of the gene. After that, I would clone the resulting PCR product into a plasmid that encodes for GFP for later detection, and then transform the created plasmid into E. coli DH5 alpha, and then induce for protein expression. Check GFP fluorescence, purify the protein, and then finally analyze through SDS polyacrylamide gel electrophoresis, or SDS page, and also the Western blot test to test for IgE binding or activity. So as I said before, my gene of interest is ARA-H1. Some background on that: there are 16 known allergen proteins in the peanut plant, but through research, ARA-H1, 2, 3, and 6 are the major ones and are responsible for activating the immune system over 50% of the time. I decided to focus only on ARA-H1 just for simplicity, and it's 1 out of 3 of the most common allergens that appear on a positive test result. So this protein is a heat-stable storage protein with a homotrimer structure, and that may contribute to its high stable binding with IgE. So I put a picture of the protein, or the protein structure, and then I found a study that extracted and purified the ARA-H1 protein, and if I were to actually carry this out, I might try their method because it's already kind of set up for me already, but they used an extraction buffer followed by ammonium sulfate precipitation, and then they elute the protein based on salt solubility. I put a little graphic of a more in-depth procedure of how ammonium sulfate precipitation actually works. The next step is fusion PCR. So basically for that, you would just need 2 DNA sequences with a region of overlapping homology. These areas will anneal to each other when you run the PCR, and they will ligate to each other. Through this method, I would attach a SSRA degradation tag, which is found in E. coli. So a little bit more about the tag. It's cleaved by CLP-XP and CLP-AP proteases. There's a few others, but those are the 2 major ones. So these proteases will detect that SSRA tag and start unfolding the protein. That protein will then be translocated into the CLP-XP or AP protease, and then is then degraded. And that whole process is driven by ATP hydrolysis. I found a lot of versions of an SSRA sequence, so I picked the one that I saw the most, and I put here the amino acid sequence, and then I just back translated it into its nucleotide sequence so that I could put it into the plasmid later. So at the bottom here, I have what my expected PCR product would look like. Next step is plasmid construction. So after looking around for what plasmid I'm going to use, which took some time, I decided to use the plasmid EGFP PBAD. It has an inducible promoter, with the inducer being arabinose. So for this experiment, I wanted to construct 2 different versions of this plasmid. So one would have the SSRA tag, and then the other would not. I did this so that in the later analysis, I could compare the two to see if there was going to be any difference. So I took the PCR product I got from the last step and then added my own primers, which had added DNA sequences for the restriction sites HIND3 and BSTBI, and then use those restriction sites to use restriction enzyme cloning to put that DNA sequence into the plasmid. You would also need the T4 ligase to, ligate the plasmid back together and seal it. So, in the next page, I have my plasmids. So here's plasmid version 1, and it has the ERA H1 protein with the SSRA tag here. There's the EGFP over here. There's also, it's not very noticeable, and it's probably because it's not zoomed in, but both of these plasmids have a his tag. That's like, I'm pretty sure when you zoom in, it's kind of somewhere in there. So that's going to be utilized later for a protein purification. So the next step is expression, and hopefully the slide makes sense. So after creating the plasmids, I would transform them into E. coli DH5 alpha, and then these would be plated on ampicillin-containing plates and allowed to grow. Colonies from the plates would be then inoculated into a broth, and then during the growth period, add in the inducer or arabinose, and then they would just be left to grow to allow enough time for all the genes to be expressed and all the proteins to be degraded and stuff like that. Once that's done, plate these again on ampicillin plates. Hopefully they have growth, and then I would check for the fluorescence. I'm going to assume that the colonies that are green under the light would be ones that did not contain the SSRA tag, meaning GFP is still intact, and then the ones that do not turn green would have the protein degraded, therefore no GFP would be observed. So after seeing the fluorescence and stuff like that, the next step would be to purify the protein. So this would be done for one for the with intact ERA H1 and one without intact ERA H1. So this protein purification can be done in a few different ways, but since the plasmid did have a his tag on it, I decided to use that method. In general, you lyse the cells, use a buffer that has a compound that binds to the histidines, and then after binding, you would wash in elute, and then you have your purified protein. Concentrations of the components will vary depending on the protein. And then one thing I'd like to note is that since the goal of this experiment was to degrade a protein, I feel like it's going to be difficult for this step to purify that protein. It's not going to be an issue with the other one where it's intact, but for the one that I hope is degraded, it might be a little difficult. But I think the next step of visualization, this step might be still okay. So after purifying those proteins, I would use SDS PAGE, or the SDS polyacrylamide gel electrophoresis. This can help visualize the protein, or like obtain masses of the proteins. So there would be a lot of different combinations in these wells. So all the way to the left, you would have, or right, you would have your molecular weight marker in one lane, then you have your controls, and then a few versions of my sample, just because I don't know what's going to happen. So I would test the purified intact protein from the last step, the purified denatured protein from the last step, and then the whole cell that's just lysed with the intact protein, and then the whole cell that's lysed without the intact protein. I do expect, if I run this, that the samples with the denatured protein would have bands that are a little bit smeared and not really defined compared to samples with an intact protein, which should have a clear, defined band. I could also do more samples in different lanes, so this would be more to help troubleshoot any issues in the future. So you could also put in one lane the sample when it was just bound to in the protein purification step, during the wash step of the protein purification step, and also the elute step of the protein purification. And these are really just more for a check, just to make sure I can go back and see, like, oh, if this went wrong or if something is not visualizing the way I expect it to, then I can see where there might be an issue. The next analysis I would run would be Western blot. SDS page does not give really any information about enzymatic activity or stuff like that. It only really determines mass. So Western blot can do that. So Western blot does use SDS page. So it uses it to separate the proteins of a sample, and then those proteins are blotted onto a matrix where they are stained with antibodies specific to a target protein. And then expression details can be determined through the location and intensity of the formed bands. So on the right, I have the result of a Western blot. This is just a random one. So on the left, you have the molecular weight marker, and then the bands, depending on the thickness of them or intensity, you can tell that there is an interaction with that antibody. So the thicker it is, the more it's interacting with it. In my lanes, I would have the molecular weight marker, and then a positive control, which is something that should react with the IgE, a negative control, which something that should not react with it, and then my 4 samples. I have the purified ERA H1 protein, as well as the degraded purified protein, and then whole cell versions, or they'll be lysed, but one with the intact protein and one without. So if I were to run this, I would expect to see a clear band on the purified ERA H1 proteins that are intact, as well as the whole cell extracts with the intact protein. For the whole cell extracts, they'll definitely have like a lot more stuff going on in their respective lanes compared to the purified ones. Since it's a whole cell, there's a whole lot of other stuff in there. But you would still be able to compare it to the other lanes and see if there's an interaction across the same row. For future directions or any changes I would make, this was obviously done in E. coli. I don't think that's very representative of human cells, so if I were to do it again, I could use mammalian expression vectors or just use parts geared towards either animals or humans. If my experiment completely failed and didn't work, I could fine-tune certain steps of the experiment, adjust to any necessary changes. I could also look at other routes, like the earlier research papers that focused on cell surface components or modifying the host. There's a lot of components to the peanut plant, and ERA H1 is not the sole protein that causes an allergic reaction, so there's definitely other factors that I could take into consideration in future experiments. And here are my references. Thank you.